So after recording lesson 6 and waiting for it to render, I don't know that I was necessarily satisfied with my description of reading frames and why there are different reading frames and how we determine the open reading frame. So I thought I would just take hopefully just a couple minutes to clarify it for you. So here we're looking at an anti-parallel complementary sequence of DNA, where I've labeled the top strand the coding strand and the bottom strand the non-coding strand. So during the process of transcription, the non-coding template is the one that's transcribed into mRNA. So our complementary mRNA would be 5', G pairs with C, G pairs with C, C pairs with G, T pairs with A pairs with U, C pairs with G, A pairs with U, G pairs with C, T to A to U, T to A. Then we'll have a G, an A, and a C, and it would end on a 3'. So here's our messenger RNA. And our messenger RNA is going to be read by the anticodons on a ribosome, and it's going to be then translated into amino acids. We have to determine where this message starts, and it's always going to start with a start codon. So we're going to take a look at this and find our potential start codon here. The first place we could start is at the very beginning of this mRNA strand. Let's see if I can't make it a little bit bigger then. Oh, sorry, I didn't mean to turn it on you guys. Hopefully, does that get everything? I think so. Shrink it just a little. Yep, that gets everything. Okay. The first potential place for us to start reading would be at the very first letter, CCG, AUG, UCA, UAA, GAC. Now we couldn't start reading here at the very beginning. That's not where our protein would start because that's not a start codon. But we do encounter a start codon right here in the second codon position. We could also search for our start codon by starting at the second letter, CGA, UGU, CAU, AAG, and then we don't have a codon. So that's our second potential place to start reading. We have a third possible place to go ahead and start reading here. We could start at the third base, GAU, GUC, AUA, AGA. Now we have to figure out, of the 3 ways that we can break all of these letters up into triplets, which one produces the longest protein. The one that has the longest protein is typically, not always, but is typically the most likely to be the open reading frame of this reading. So we said in our first reading frame, the green reading frame, that the first one is not a good potential reading frame or a good potential place to start because it's not a start codon. But if we make it to the second one, there's 1 codon, 2 codons, 3 codons, 4 codons. So it's possible that we have a strand that would be 4 amino acids long here. If we were to look at our second reading frame, CGA is not a start codon. UGU, not a start codon. CAU, not a start codon. AAG is also not. So we don't have a start codon in our second potential reading frame, making it exceptionally unlikely that that's our reading frame. If we then take a look at our third reading frame, we have right here GAU, that's not a start codon. GUC, that's also not a start codon. AUA is not a start codon. and AGA is not a start codon. So in this particular strand of DNA that we've been given, if we're reading the top strand is the coding strand and the bottom strand is the non-coding strand, there's only one potential open reading frame. Now, if we take away my hypothetical that the top was the coding and the bottom was the non-coding, and we reverse that, it's possible to read this strand in the opposite direction then. So rather than reading the black strand as our potential gene, we're going to read the red strand as our potential gene. But because DNA is anti-parallel, we can't start at the beginning right here because this is the 3' end. So we have to start reading down here. Now I want to point out just, small things. If we look at our gene, you'll notice it says ATG where there was a start codon. So as we look at our potential template, we're going to look at it for where it says ATG. So it might be easier for me to start a new slide without all this writing on it. So I'll do that. I'll flip to that slide I accidentally created. Top strand, like we did on this first slide, we would have we'd have 4 amino acids, so a protein that's 4 amino acids long. AU. So taking a look at this slide then, remember we're going to reverse our assumption from the first slide, and we're going to pretend that this is the one that we're going to read instead, starting at the right because we have to start at the 5' end for our reading. Our first potential reading frame gives us that as a codon, and I'll just mark our codons as we go down. So we have one potential reading frame, and then our second potential reading frame of this strand and our third potential So if we then try and do the same thing we did before and determine how many codons each of these potential reading frames is, if we look at the first codon, we are looking for ATG, because remember this is DNA, not mRNA, so it would be AUG is what we want, but since it's DNA, it's ATG. Our first codon is not a start codon. Our second codon is not a start codon. Our third codon is not a start codon, nor is our fourth or fifth. So our first potential reading frame on the complementary strand has no start codons. Our second potential reading frame, 1, 2, 3, 4, again, no start codons. Finally, in our last potential reading frame, we have CTT, no potential start codons, but we have an ATG here in the second position. So that would be our first codon, second codon, and third codon. So of the 6 different ways that this strand could be read, if we read the bottom strand, as we did on this particular slide, we would have a protein that is 3 amino acids long. Remember, the open reading frame is the one that has the most, or is typically the one that has the most amino acids, which means it seems like the top strand is in fact our coding strand, and our open reading frame is that first reading frame that we looked at. Hopefully that makes a little more sense. I tried to slow down a little bit more and highlight it a bit better. That's what we do when we look for open reading frames. And that's what we've created algorithms and search programs to find for us, is how do we maximize the number of amino acids in any given particular section of DNA that we know is a gene. All right, hope that helped. If you guys have any questions, you know how to find me.